Welcome everyone to this SynCell 2021 speaker series. My name is Telmo Diaz. I'm a Ph.D. student in the University of New Mexico for Dr. Carol and Dr. Lopez lab. And we have two very interesting talks today by Dr. Felipe Garcia Quiroz and Dr. Cesar Rodriguez Emenegger. Remember that this is also streamed on YouTube. So if you want to access later to the content that today is going to be live streamed, you're going to be able to do so. If you are joining by Zoom, please submit your questions in the Q&A section. And if you are joining by YouTube, you can use the comment section and we will transmit the questions to the moderators. So first of all, we have Dr. Felipe Garcia Quiroz, who was trained as a biomedical engineering in his native Colombia, to later go to Duke University and obtain his PhD from the lab of Ashutosh Chakori. He focused on his research about self-assembling protein for biomedical applications. A very important outcome of his PhD was the elucidation of sequence rules that rationalize the ability of intrinsically disordered proteins to form membraneless organelles in the cell. And after his PhD work, he moved to the laboratory of Elaine Fuchs at Rockefeller University, where he did a postdoctoral fellow when that led to the discovery of ganglionic repeat proteins in skin assembly, a surprisingly extensive network of membraneless organelles with unique liquid-liquid properties that are crucial to the process of skin barrier formation. Felipe is the recipient of a career award at the Scientific Interface from Borough Wellcome, and he is now an assistant professor at the Georgia Tech and Emory University. He has his own lab, and his research engineers self-assembling materials that are genetically encoded and stimuli-responsive. He uses his research to challenge some features in the fields of nanotechnology, biotechnology, and medicine, and his results illuminate fundamental aspects of biology that serve as foundation to engineer advanced biomaterials. And we thank Dr. Quiroz to be here with us today, and thank you for being very active in our speaker series. So I'm gonna let you share the screen now and take over.
Thank you, Telmo. Do you see my screen?
Yes, all good.
Thank you for that very nice introduction. And my team is fascinated by stimuli disordered proteins that over space and time form very defined structures in the cell. And so I'll be talking about these membraneless and stimuli-responsive organelles. But we have a deep interest in self-assembly in general, particularly, again, thinking about disordered proteins or IDPs. And if you look at the molecular level, you always see a different picture for these kinds of IDPs, which is unlike the kinds of rigid structures that we typically think of when we think of proteins like helices. And so over the years, we've done a lot of work looking at how to map the sequence space to understand fundamental principles of about how these proteins behave, particularly in the context of how these proteins then self-assemble into structures like the nanorods shot in this image. And so generally, we're thinking about, again, a transition from a soluble state of these IDPs in a stimuli-responsive manner into some sort of aggregate or compartment. And more recently, we've been thinking about this process in the context of cells and tissues. And the examples that I'll be showing you today are all really inspiring our work in the concept of skin, where we just demonstrated that these phase separation processes happen in the cells of the skin and are very important for the formation of this of the skin barrier. So what is going on in the cell then? So you can think of the cell, of course, as a mixture of proteins, but if one of these proteins has the ability to undergo any of these kinds of phase transitions, if the conditions are met, one of these proteins would then mix from that complex mixture, forming its own compartment, even without the need for a membrane. And so you can map that to a phase diagram as you would in polymer physics. And so you have protein concentration and a given temperature or some sort of stimuli, but if the conditions are met and you pass that critical concentration for a given temperature, the system will then mix into a high density phase, forming the scaffold, the core of that organelle, and a low density phase that would be sort of what's left in the cytoplasm. So this polymer physics framework gives us a way to begin to look at these kinds of organelles. But they're coming of age. For a long time, of course, we learned the cell is made of membrane-bound organelles, but for a long time, we've known that things like the nucleolus are there and they don't have a membrane. But over the last decade, there's been an explosion of examples of very intriguing organelles that are membraneless, that have interesting biological functions. And here's just a list of examples, all the way from RNA processing to chromatin organization. And more recently, in the context of tissue biology, so multicellular structures, where tight junction assembly is being shown to be related to phase separation behaviors. And in our own work, as I'll show you, skin barrier formation. And as we learn more about these kinds of organelles, now there's opportunities for engineering them. And my favorite example, and there are not that many yet, but my favorite so far is one in which the group of Edward Lemke in Mainz, Germany, created an organelle for orthogonal translation in the cell. Beautiful example. Here's a citation. So what are these properties then that can be controlled by phase separation? And so you can think of any of these proteins, of course, as subject to post-translational modifications in the cell. And if one of these modifications favors interactions, then you can drive assembly of these organelles. Likewise, and conversely, if you have an organelle and there are post-translational modifications that counter any of these interactions, then you have this assembly. And because these are very dynamic interactions controlled by these phase separation dynamics, they're kind of basically liquids. And so depending on their viscosity, if any of these condensates in the cell, in the cytoplasm would meet, they can readily fuse, and this would depend on the viscosity of these structures. But also you might have different kinds of condensates or membranous organelles, and their relationship as they meet will depend on their surface tension and their affinities, so that they might either fuse forming a new organelle, or they might actually remain as distinct organelles. But the view that I just offered is one that is kind of dominated by polymer physics, very homotypic. It's almost like thinking as the cell as a test tube where you only have one protein that undergoes these kinds of phase transitions. However, of course, the milieu in the cell is much more complex than that. And so you can immediately begin to think of even just three proteins that are multivalent and have an interesting interaction where they have different affinities for each other. And the work of Mike Rosen and others has, I think, made an important contribution demonstrating that beyond IDPs, if you really just think about multivalent interactions, any of these scaffolds, if they meet a multivalent scaffold for which they have an affinity, they can potently drive to the formation of membraneless organelles. But because these organelles have binding sites available for other proteins to join, these so-called low valency clients might get recruited to the membraneless organelles, adding functionality to them. And so it serves as a framework to think of these membraneless organelles as having scaffolds, those that drive assembly and maintenance of the organelle, and clients, those that are just there to add functionality. So with that in mind, I wanna sort of pause for a little bit and tell you then what I want to talk about for this talk. And so, of course, I want to inspire you as a community thinking about synthetic cells, about how these membraneless organelles might be important to your work. And so I want to discuss aspects of their assembly or disassembly, particularly how it's governed by phase separation dynamics, how might even the crowding of the intracellular space might be possible with these kinds of organelles that are supposedly essentially prone to fusion, what their stimuli responsiveness is like. And importantly, as we think of any organelle, you gotta control its functionality and composition. So I want to show you examples of how we can readily control and target components to these organelles, what kinds of mechanisms can be actuated by these sort of membraneless organelles. And lastly, I want to touch briefly on our own efforts to think about how we can engineer these sorts of membraneless organelles. I'm going to accomplish this hopefully by telling you about one system. And so that system is, as I said earlier, is our own work in skin biology. And so I need to introduce the system first. So here's the skin. It's a multicellular structure. So follow me here. We begin with stem cells at the bottom, and they're bound to some extracellular matrix, a basement membrane, and they differentiate by moving upwards. And so they move upwards, and as they do so, they acquire new proteins, and so new unique structures. And particularly right below the skin surface, there is a layer called the granular layer, and it's called granular because of these red granules. They're called keratohalin granules, and they've been known for 100 years, but we knew very little about them. But to us, now with this new lens of phase separation of membraneless organelles, like, oh, wow, this may be really intriguing and put understood epidermal membraneless organelles. And so we began to ask questions about, okay, well, what are these structures like? Are they liquid-like, as one would expect, or are they solid? How are they interacting with the nucleus and other membrane-bound organelles? Again, there is an intriguing transition that has to happen for the skin to form, which is that this normal-looking cell having a nuclei, mitochondria, and others has to lose those membrane-bound organelles to form this so-called dead surface of the skin. And so how is keratohalin granule disassembly perhaps contributing to that process? These were the kinds of questions that were really driving a lot of our thoughts at the time. But as you'll see, and hopefully I convince you, this provides an interesting framework to understand how membraneless organelles function and how we might engineer them for synthetic cells. We had a clue, and this is how the story begins. We knew that a protein known as FLG or filaggrin was abundant in those structures. And so here is just an immunofluorescence image of mouse skin showing the localization of that protein, very abundant in the skin of the mouse and always localized to those particular granules. So what is this filaggrin? And when we looked at it, it was obvious. It was a prototypical phase transition IDP. And so let me walk you through it. It's an intrinsically disordered protein made of multiple repeats. And I know that it's intrinsically disordered, but I can show it to you with the disorder plots. 0 is ordered, 1 is disordered. And so you can see the entire length of this gigantic protein is essentially at 1, so disordered. Except for the S100 domain, this blue domain at the N-terminus is a dimerization domain. But if you actually look at the specific composition of that protein is so-called low complexity, because only a handful of amino acids make most of its composition, particularly histidine, serine, and glycine, which we knew were important for phase separation behavior. And then if you look at its size, these are among the largest proteins in the human and the mouse proteome, which we knew was important for multivalency. And then when we look even closer, some of the unique features of this protein, like the fact that it would never have a lysine, but rather prefer an arginine, was a sequence bias that we knew was important for phase separation. So again, all pointing to prototypical phase separation behavior. Now, if you actually look at these repeats, they're identical almost, 95 to 99% identical in composition, in sequence. And so we began by basically taking a given repeat, because they're essentially all identical, and began to build constructs with different number of repeats to probe this phase separation behavior in the intracellular space. And at any time throughout this assembly process, we could add fluorescent proteins, we could add different domains to begin to probe the functionality of these proteins. Let me just give you an example. So this is a fixed concentration. And here I have 2 constructs, one with 2 repeats of the RA domain. And you can see the protein is essentially diffused in the cytoplasm. But for the N equal 8, so 8 copies of the repeat, now you see very well-defined formation of what appear to be membraneless organelles. And we did this quantitatively over a wide range of expression levels. And so here's the quantification, where for N equal 1 or 2, it doesn't matter how much you make in the cell, it doesn't undergo phase separation. But for 4, 8, and 12, you begin to see sharper and sharper phase transitions, so that for full length, we estimated that about 1 micromolar is sufficient to trigger a very well-defined phase transition inside the cell. Now, how about their liquid-like behaviors, however? And we can probe this using photobleaching. And so here is one of these compartments in the cell, and we can photobleach it. And just 13 seconds later, there is recovery. If you quantify that, so photobleaching happens, you lose fluorescence, there's recovery, is very fast and complete. Now, of course, this will depend on the size of these proteins. And so as these proteins get smaller, you also see that the recovery is happening faster. So the liquid dynamics are changing. Nonetheless, they remain very liquid. Now, if you do live imaging of cells in culture, you begin to see that over time, when these organelles meet each other, they undergo liquid-like fusion events. They drop like droplets of water that are coming together. Now, this is cool and in line with what pretty much everyone has seen in the context of membraneless organelles, but to us, it was like, wait a second, this is not maybe so cool, because if you remember from earlier, in the context of the epidermis, these organelles appear to be crowded in the cytoplasm. So if they're liquid-like, and we now see that they do undergo liquid-like fusion, what is going on in the epidermis? How is that happening? And that was also cool to us, and to me particularly, because I knew that it was telling us something about a challenge that we will have when engineering membraneless organelles, which is that you may not be interested in having one single organelle. You may want to have many coexistent. And so perhaps there is a solution that is offered by epidermis. And I'll show you our efforts to uncover this. But to do this, I need to tell you what we did to then examine the endogenous organelles in the skin. And so instead of building our own engineered organelles, we wanted to actually look at the ones in the skin. And so to do that, we thought, well, why don't we just target something that is some sort of fluorescent sensor to those endogenous organelles to be able to study them? And the rationale was very simple. Well, if you were to regulate a protein that undergoes phase separation in the cell, like filaggrin, it would clearly form its own compartments. But I would like to have a sensor that is already present in the cell, and that if experiencing the presence of this other phase separation protein, that it would then report on that phase separation behavior. And the rationale was very simple. IDPs with sufficiently weak and phase separation specific interactions should be able to report phase separation behavior. That was the rationale. So let me show you how it works. I already told you that one repeat from filaggrin is not good enough to drive phase separation behavior. So what you can do is you can tag it, right, with just GFP, for example, and then co-express it with filaggrin. And so I'm going to show you what that looks like. So here's a cell that has filaggrin, so it forms condensates really well. And here's the putative sensor. And you can see that it does show the localization of the filaggrin. So the condensates are shown, but the partition coefficient, and I'll be using this consistently as P, the partition coefficient is just twofold. So it's just twofold enriched in the condensate. It's not really that great. So we began tuning the specificity of these interactions to enhance them, while at the same time playing with different domains that we know would also affect this phase separation behavior. I ended up, keeping the story short, two sensor designs that are very different and actually unlike the filaggrin sequence, and among themselves are also very different. And let me just show you what it looks like. So again, here's a cell that co-expresses filaggrin, forms condensates or these apparent membraneless organelles, but the sensor is also expressed, and you can see now that the partition coefficient is very high, about 25. So it goes exquisitely well into the organelles, unlike the first design. So now we have a tool that seems to be targeted really, really well and efficiently to the organelles. Of course, I need to show you a control, which is that if filaggrin is not present, these sensors do not form compartments of their own. So these sensors are indeed soluble in the cell, and only upon the presence of this phase separation protein show and report that phase separation behavior. So with this tool, being a genetically encoded tool, we could put it and package it into lentiviral vectors. And these lentiviral vectors can, express our constructs, these phase separation sensors under the control of a given promoter of interest. And we can also play additional tricks like adding hairpin RNAs to knock down any protein of interest. And I won't show you a lot of examples of this because of time, but it's a tool that we use extensively in our studies. But this is more or less what it looks like. You create these, you inject these viruses in utero and are able to very quickly create these genetically engineered mice so that the surface of these mice, their skin is tagged with these fluorescent proteins. And as I'll show you later, this basically allows us to then begin to look at these phase separation dynamics in the skin directly. And so this is a cross-section of what the skin looks like. Again, basal layer all the way to the corneum, as I showed you earlier. And if you look at the signal from these sensors, you see that there is granularity appearing as you move towards the top being the cornea, right? Where the granules would be expected to be seen. But if you now look at planar views, you now see something really remarkable, which is that in the early granule cells, you have very few of these granules and they're very small. But as you move towards the surface through the middle and late granular layers, you begin to see very large granules. Consistent again with what we had seen actually just from immunofluorescence. So the sensors seem to be showing us what we wanted to see. But how about the dynamics? This is, of course, something that we can do through live imaging. And what we saw, again, to our surprise, but consistent with the immunofluorescence, is that over the span of about a day, if you do imaging of the skin of these mice, there's a lot of growth of these granules, but there's no fusion. They're grown without fusion. And we can quantify this. And so if you look from 0 to 800 minutes, there's a lot of growth. You see that new granules, new compartments emerged, some grew in volume. In fact, if you quantify this, some of them are growing by about 10-fold in volume. So a lot of growth, but no fusion. And so to us, this was really puzzling because, what was going on? Maybe these characterizing granules in the epidermis are not liquid like we thought from our in vitro experiments. And so one thing that we can do, of course, is photobleaching again. And so we can take these sensors and we can photobleach them. And you can see that in 2.5 seconds, there is recovery. And this is sensor A, but this is now sensor B. And we basically see the same. This is the quantifications on the right. Within 2-3 seconds, there is recovery, right? So these compartments are mixing their components very rapidly, indicative of a liquid-like behavior. So what is going on there? And so this forces us to go back, because in our initial experiments, when we did the experiments in vitro that I showed you earlier with extensive fusion, we were working with a cell that was unlike the cells in the epidermis. And one major characteristic of the cells in the epidermis, particularly in the granular layer, is that they have acquired keratins that are very specific to that stage of differentiation. So we went back and we thought, well, maybe this cytoskeletal network that is made by the keratins in the epidermis is somehow, adding an additional layer of structure to the cells that prevents fusion. And so we created these engineered cells that will have filaggrin compartments but also expression of a fluorescent label keratin that's specific to a granular layer. And we began seeing that whenever we would see any keratohyalin granule, we would also see cages. It seemed just like the keratin was kind of surrounding very nicely these structures. And when we then did live imaging experiments, we would see that, you know, in regions of the cell with very low keratin content, you would actually see nicely emergence or fusion of these compartments of these filaggrin granules. But there would be regions of the cell with lots of keratin, in which, again, the granules were kind of embedded in this keratin network. And then we wouldn't see fusion unless they would detach from the keratin network. And in even more striking examples, we would see cases where we would see two granules that are adjacent to each other, but there is like a thick fiber in between them that is acting as a fence. And then we will never see fusion. And this is best actually seen with an optical section. So now you see these basically two closely interacting granules. We never see fusion, but now we understand there is a fence in between them that is preventing that fusion. So that forces us to rethink a little bit of the keratin part. And so here is the structure of keratins, and particularly human keratin 10, which again is very abundant in the granular layer of the epidermis. And one part that is poorly understood about these keratins is they actually have N and C-terminal IDP domains. So we thought, well, maybe these IDP domains are interacting with the granules and with filaggrin. And so we created an in vitro experiment in cell culture where we could create granules, and you can see that M. cherry, for instance, is very well excluded from these filaggrin granules. The enrichment is actually 0.4, so it's really not enriched at all. It's actually excluded. But if you add the IDP domains, the N and C-terminal IDP domains from keratin to M. cherry, now they go into the compartment. Now the enrichment is about 1.6. So the N and C-terminal domain for keratin 10 from the experiment we concluded is actually allowing for interaction with the filaggrin in the condensates. And so to keep the story short, what we ended up doing was going back to the epidermis and creating genetically engineered mice with fluorescently tagged keratins. And it allows us to visualize the maturation of the keratin network in conjunction with the maturation of the granules. And we saw consistently that the crowding with keratohyalin granules was hand in hand with keratin bundling. So that by the mature granular stage, you would actually have a very mature keratin network surrounding the keratohyalin granules. And this is well seen in this cross-section on the top, on the right of this image, where you see that the granules are in fact embedded in keratin cages. And so the keratin network is in a tug of war with the keratohyalin granules to allow for this massive structuring and crowding of the cytoplasm that we see in the epidermis. Now, of course, you know, we wanted to know the functionality of these structures, and this gets to stimuli responsiveness, and you'll see that in a bit. But essentially, I want to go back to the epidermis. And so I told you that one of the intriguing things about the epidermis is that when you go from this layer in the granular state to the corneum, the surface of the skin, the cells change dramatically, right? They have to lose their nuclei, they have to lose their mitochondria and organelles, and they do that very rapidly. In our experiments, we saw this happens in about 2 hours. So very rapid process, poorly understood. Could it be that the keratohyalin granules are playing a role? So we began doing imaging experiments where we combined imaging of chromatin and imaging of our sensors to mark these keratohyalin granules. And we consistently saw beautiful coordination of the process of compaction of the chromatin, which is the initial stage of enucleation, with the disassembly, a partial disassembly of the keratohyalin granules. And we, with experiments that I don't really have time to show you, we demonstrated that that's critical for the process of enucleation. But what I do want to show you is that upstream of this keratohyalin granule disassembly and enucleation is a beautiful example of stimuli responsiveness in membraneless organelles. So we knew that the surface of the skin was acidic. And this is well known for a long time. Your skin is about 5.5 on the surface. But that's extracellular pH. We, of course, are dealing with intracellular organelles. And so we thought, well, maybe there is a transition somehow around the granular layer that maybe there is a extracellular pH around 6.5. We know that in the basal layer, this is normalizing to 7.4. But intriguingly, if there is transition in that pH gradient, this is interesting because filaggrin is a histidine-rich protein, and histidines have a pK of about 6.1. And we knew that would alter phase separation behavior. So we began doing experiments in vitro where we would co-express filaggrin to form condensates in cell culture together with sensors. And so you can see this perfect co-localization of the two. That's why they look white. But if you change the intracellular pH now to 6.2, now you go from beautiful co-localization in granule 1, granule 2 to partial disassembly of the granules. This is seen in the magenta signal for filaggrin, and then in the green signal for the sensor. So there is a pH-actuated disassembly of the granules that allows for the release of these components from within them. And we know that this is specific to the PK of histidine because when we did experiments at different pH values, it's only once you go down to about pH 6.3 that you begin to see prominent release of the filaggrin signal. And suddenly, if you keep going down to more acidic values, you actually get more prominent disassembly. So we thought this was a really cool example of an actuated process in a pH-responsive manner to trigger disassembly of membraneless organelles. And I don't have time to show you how we went back to in vivo to demonstrate this process in the context of the epidermis, but I want to kind of summarize what we found. So this is kind of an overview of what the skin looks like from the perspective of phase separation. And so early on, when the granules first appear, there are very few keratins there. So, when granules meet, they do undergo fusion. We see examples of that in our live imaging experiments. But once you get to the middle granular layer, there is crowding that begins to happen because the keratins are forming cages around these granules. And as they do so over the span of about 1 or 2 days, these granules grow substantially as the keratins also bundle. When they get to the critical stage, like this example of this cell that is about to transition to a corneum, there's a massive disassembly process that happens that is, as I showed you earlier, pH triggered. And what we now know that happens is that this intracellular pH shift is again leading to disassembly of the granules, triggering compaction and completing that process, leading to the formation of early cells in the corneum, these so-called squames. And so what I'm not showing you, however, is that if you remove these compartments, even an intracellular pH shift is not sufficient. So we know that this is a keratocyte ion granule dependent process, that the sensing of this pH shift is done by these keratocyte ion granules, and that there is a prominent release of components from within them that we think are important for enucleation. There that I have to show you, there are very intriguing interactions between the organelles, these membraneless organelles and the membrane-bound organelles, like the nuclei, for instance. We see the formations of those organelles. We know they're important, but we don't know the role in detail. And so there are gaps that we still have. Like, we don't really know what these clients, these components of the keratocyte ion granules that are released in a pH-responsive manner to actuate enucleation are still unknown to us. But there are examples, these are things that we can target in the future. And so I want to go back briefly to this idea of targeting to the compartments. And so you might recall that in the context of multivalency, you might have clients that get recruited, right? So if you know a binding domain, you can deal with that. And so let me give you an example. We know that there's an S100 domain, it's a dimerization domain, so we can create that, and we can create a client that has a S100 domain to allow for dimerization. But look at this. It does go into the compartment, but it does it only with a partition coefficient of 1.3. Of course, if you remove the S100 domain, then you have no partitioning whatsoever. So you can definitely take things to the compartment that way, but it may not be as efficient. Now, you may introduce your own domains of interest. So in this case, we created one that is completely engineered. It's a cleavage site of the tobacco virus protease. And so now you see that with this new domain added, now this protease that is dead at the very end of it can go to a compartment, but the partition coefficient remains low. However, it's specific. If you mutate the cleavage sequence, now the protease actually does not localize. But I want to put this in context of the phase separation sensors that I alluded to earlier. So again, partition coefficient of this protease is 2.1. For the sensors that I described earlier, the partition coefficient is about an order of magnitude higher. And importantly, it does not depend on the concentration levels. And so for very low to very high expression levels, you still have a very consistent partition coefficient. And more importantly, because it's not a one-to-one binding, you can actually accumulate a lot of these components in those structures. And so much higher amounts within the granules than what you will get with this one-to-one binding. And because it's not a one-to-one binding, we don't see that the presence of these components within the compartments alter the dynamics. So this is FRAP data for the scaffold filaggrin. When in presence of the sensor, we see that the filaggrin doesn't care. But when you have a client to which that binds to the scaffold, now is dragging on the scaffold, and we see that the dynamics are altered. And this is because the scaffold, the client actually does bind to the scaffold with sufficient strength. Whereas if you compare that with the sensor, the FRAP is so fast that it's indicating that the affinity is just really, really low. So you can accomplish beautiful targeting to the compartment despite these extremely weak interactions, which we think is remarkable. Now, what do you do with that? And so we began to think about restricting reactions to the keratocyte ion granules specifically. And so here's an example, just to give you a proof of concept. And so we're restricting bioattenuation. So what we're doing here is taking one of these sensors and attaching at the genetic level peroxidase, and this is Apex2. And when you provide a biotin phenol to this reaction, and this is mouse skin, this presence of the sensor now is sufficient to drive bioattenuation, but the bioattenuation is specific to the compartment. And you can see this from the merged image. Now, if you leave out the biotin, this is a biotin phenol, again, you see at the triptavine labeling, there's no triptavine labeling because there's no biotin to label, right? So we're really restricting now this bioattenuation reaction specifically to the condensate. That's important to us because this bioattenuation can serve as a proximity-dependent tool for us to then purify the components that are tagged with biotin to begin to ask who are those endogenous clients that are maybe responsible for the biology and for the functionality of these keratohalline granules. Of course, this also shows that we can restrict reactions to these abundant organelles of the epidermis. And I want to finalize, this is just nearing toward the end of my talk, with the idea that we have a strong interest in engineering synthetic versions of these organelles. Why? Because I didn't have time to show you, but we actually know that this protein, gigantic protein, is heavily mutated in humans. And we actually did a lot of work to show that these mutations, which are nonsense, so truncating mutations, they potently prevent assembly of these organelles. And we know that and we've been able to link this to skin barrier disorders. So could we shrink this gigantic protein, which is, really not possible to work with in the context of trying to, to rescue this process in a realistic setting, to a mini filaggrin, so a mini version of these proteins. And so this is drawn to a scale, basically, where we could take one of these domains, so R8, for example, again, and we'll create variants of it that have different phase separation propensities. And we do that in two ways. One is just basically playing with terminalization domains or multimerization domains, in this case, NC1 from collagen 18. But you can also mutate. So there are SNPs in filaggrin that would change from a histine to a tyrosine, which we know would promote phase separation. And so we begin to create variants adding terminalization domains and adding these non-SNPs. And I just give you a flavor for that. If you create trimers of just R8, that's sufficient to drive the assembly of some sort of phase separated compartment in the cell. But they're very low viscosity compartments. And you can see they're basically just flowing in the cytoplasm. But when you go to the other variants that we created, you begin to see very well-defined organelles that are actually more reminiscent in viscosity to the endogenous compartments. And in data that I didn't have time to show you, we want to tune that viscosity because we know that it's important to the functionality of these structures. And just to give you a flavor for this, we began to now put these constructs back into the skin. And so we can deplete the skin of the mouse from these endogenous compartments, creating basically a new canvas. And we can now add in our constructs and we can begin to see these engineered organelles in the mouse epidermis. And so we're doing these experiments to basically understand how these engineered organelles might be able to rescue some of the skin barrier defects that we've seen. And so with that, I just want to sort of wrap up and get you thinking about membraneless organelles in the context of synthetic cells. And so I hope that, from my talk, you get the sense that there are multiple avenues for controlling the composition of these organelles, that even if we're talking about liquid-like behaviors, I mean, they can span a wide range of material properties. And that phase separation dynamics is basically the framework to program the dynamics of these organelles. And because of that, basically, you can think of these organelles as stimuli-responsive compartments, stimuli-responsive materials, in fact. And now thinking about the future, there are a couple of things that I want to highlight. And so I think the time is right for sure to create designs of membraneless organelles for the synthetic cells. We have all the elements required. There's exciting opportunities in the context of multi-phase architectures, like the one shown here, where now we have liquids within liquids. And some groups have made progress in that direction. We're very intrigued by membraneless organelles. Basically, can you have a mixture of these organelles coexisting, and so that you assign different functionality to different organelles? And in an area that we're really invested in is the exploration of non-equilibrium phase separation dynamics. And this is important because a lot of the examples that we know of are essentially in equilibrium, right? You basically undergo disassembly and assembly in what appears to be an equilibrium process, but we know that IDPs and many of the proteins implicated can engage in behaviors that are far from equilibrium. And so we think that exploring that avenue will give us a new handle into controlling the architecture and controlling the dynamics of these membraneless organelles in ways that it could open up interesting avenues for new functionality. And so with that, I want to thank this work that goes back to my time at Duke with Toshio Cotti, collaborators at NC State, A lot of the work with the epidermis happened in the lab of Elaine Fuchs at Rockefeller University. I'm very thankful for that. And in my own group, Alexa Vesilla, one of my PhD students, contributed to several ideas and pieces of data in this presentation. So I thank her for that. And I'll take the opportunity to advertise. And my group is very young, and we're actively recruiting. So if there are students there who are interested in postdocs or research scientist positions, please reach out. I would love to hear from you. And with that, I'll take questions.
All right. Thank you very much, Dr. Quiroz. That was a beautiful talk. I'm going to go ahead and read you some questions. The first one from Jacqueline Delora. She says, beautiful work, Felipe, especially the design of the sensors. To clarify, do you expect that the differentiation of the cells found in each skin layer is regulated by the liquid phase separations of filagrins?
Thank you, Jacqueline. Yeah, it's a great question. This talk, I tried to sort of like keep it very light on the details of the epidermal biology, but we invested heavily in linking the phase separation dynamics that we saw and the biology. And we see that at two different levels. There's a biophysical component, and then there is a biochemical component. Biophysical component is one that relates to the crowding of the cell. So we actually see that because there are so many of these membraneless organelles, that ends up being important to the structuring of that cell as it goes towards the skin surface. And we've seen that that's important for the mechanical properties of the epidermis. And biochemically, although we don't have the specific lines that are being released to actuate enucleation, we definitely have seen when we deplete these compartments that there's no pH responsiveness actuation of enucleation. And so we definitely have shown that it's essential to have these dynamics. So the assembly and disassembly is important. It's not just the formation of the organelle. In this context, it's actually the disassembly that then plays a major role in releasing these key components, which again, as I said, remain to be defined. So we're very excited about the proteomics work to begin to actually parse out the specific components that are responsible for that. But there is definitely links between assembly and disassembly, and they're biophysical and biochemical.
Right. Thank you very much. Gabriel Lopez asked a question. He says, fantastic talk. The changes in microstructure of cells as you go through the granular skin layer must affect the cell's mechanical properties. Can you correlate cell mechanical properties to pH in vitro in a way that is relevant to mechanical properties of the skin cellular layers?
That's a really, really cool question, Gabriel. We've done a fair amount of thinking. We've been using AFM to probe the mechanical properties of these organelles. They're essentially mechanically responsive as well, as you can imagine. We know less about the details of how the crowding, particularly cytoplasm, is altering the properties of the epidermis itself. But when we do experiments in vitro with granules that we would expect to have different viscosity, we see different we see different mechanical responses of the cells. And so you say have a low viscosity compartment versus a high viscosity compartment, and you begin to see the cavities, of course, going there as well. So you get reinforcement of those regions of the cell, and we can see that by AFM in cell culture. The prediction is that that translates to the epidermis, and I think it's fair to say that it probably does, but we don't have data to show that. So I do think that it's really important to think about it, especially because the skin has remarkable elastic properties that need to be controlled and are probably related to the structure of the cytoplasm. So it's things that we need to think about more, but at least the evidence that we have, we can think of these membraneless organelles as mechanically responsive. And so they do play an important role, I think.
Great, thank you. So Ao Fen Zeng asks, I am a freshman in college, so a lot of this is very new but interesting to me. I'm interested in the medical or practical applications of creating synthetic membraneless organelles. He wants to know a little bit about those applications.
Yeah, I mean, it depends. It's certainly a fundamental question. It depends on the biological system. Many different kinds of applications. In the context of the skin, as I alluded to towards the end, our major driving force is that these mutations in the major scaffold that forms these compartments in the skin is heavily mutated. And so particularly in Asians and Northern Europeans, these mutations do create a lot of trouble. They essentially prevent assembly of these organelles, and they're known to be linked to things like ectopic dermatitis and in some cases psoriasis. And so what we're interested in is, can we create synthetic organelles that would rescue those skin barrier defects, right? They're lacking these organelles. Can we create a system that would rescue that? We know that it's hard to do it with endogenous protein, as I said, because it's so large. Technically speaking, it's almost impossible to work with such gigantic proteins. There's no way to deliver them. So we think that by shrinking them, but still recapitulating the formation of those organelles, we might have a shot at rescuing or sort of curing those disorders. In fact, the therapies that exist out there to treat those disorders are palliative in nature. What you're doing is you're dealing with the barrier defect by damping down inflammation. So immunosuppressive strategies. What we think is possible now is to rescue the underlying barrier defect, which is rooted in the lack of these organelles. So at least in the context of the skin, there's a clear application, and there's fascinating stuff in the context of neurodegeneration. So as a freshman, I encourage you to look at neurodegeneration. There's beautiful, beautiful opportunities for work in that space as well.
All right, thank you. Another question is related to the experiments about the disassembly of granules within pH. When you change the pH gradient to disassemble these stress granules, can you have a fully disassembly, or there is a degree of hysteresis?
Oh, that's a really cool question. So you may have noticed from the images, when you shift that pH, it's basically impossible in our system to complete this assembly with pH. The reason for that is that you essentially have millimolar amounts of a pH buffer. And so even though you get to trigger the initial stages of this assembly and there are changes in phase separation dynamics, over time, there is pH buffering that happens. And so we've seen this in different ways as pH reporters. So we think that what's going on in reality is that the pH trigger is the initial stage of this assembly that changes the permeability of the organelles and allows for other components to enter, like enzymes that add PTMs, post-translational modifications, that complete this assembly. So we think actually pH is not sufficient, which is it's a great catch, but that the changes in permeability then now enable the later stages of this assembly, which probably we think involve PTMs, and we've done some work in that space. We have implicated phosphorylation particularly. So the kinases, we don't know who they are, but we know phosphorylation is a potent way to complete this assembly. Now, hysteresis is a whole different topic. I don't even want to go there.
Thank you. All right, a couple of last questions. Idira Matthias asks, great work. You mentioned that the release of components is important for nucleation. What components are these, and how do the compartments change?
So yeah, unfortunately, we don't really know because we haven't gotten the proximity epidermics data yet. But you can imagine things like nucleases, right? We know that the chromatin needs to be cleaved. And so we think that most likely there are nucleases, that DNases, for example, that are trapped in the condensates, that they might be released in a pH responsive manner to accelerate the process of enucleation and chromatin cleavage. But we don't really know. And so what we're doing is, with the strategy that I show you, where we can biotinylate the components of these granules, what you can do now is you can break the skin, you can lyse it, and just literally pull down on the biotin-labeled proteins, and in that way, build a map of who those components are. And so we think that this biotinylation strategy, which I sort of presented more from the perspective of, hey, proof of concept that you can restrict reactions, in reality for us is a major strategy to then answer the question that you asked, which is, hey, who are those components? But we suspect that nucleases, and other proteases as well are probably implicated.
Thank you. And last question from Jack Holland. Do you know if the KG caging by keratin is ubiquitous amongst the higher organisms? And do you know if the KGs co-evolve within the keratin network?
That's a really cool question. And for the most part, I don't really have a good answer to it. The evolution of a lot of these sequences is pretty hard to track because they're very low complexity. And they evolve very, very rapidly. And I don't really have a good handle of how these IDP domains in keratins particularly evolve. I can tell you one thing, however, the keratin family is very large. There are 70, something really, dozens and dozens of keratins. And there is a lot of variability, particularly in the IDP domains. As I said, they evolve rapidly. And so even for the epidermis, in the mammalian epidermis, we don't have to go very far. If you compare the keratins that are found in the basal, so the stem cells at the very bottom, those keratins are different than the ones in the granule layer, and they're different mostly at those IDP regions. And so, you know, we know that they're specific, so there's probably some sort of coevolution, I guess, in that sense. They're really kind of answering specifically to the interaction that you need with those keratin, between keratins and filagrins. But I wouldn't go as far as maybe thinking beyond mammalians. But at least in the context of the epidermis, there is a lot of specificity to these IDPs that are basically interacting with the granules in a way that we don't see for our keratin. So we do think there is some sort of interesting interaction coevolution there, but not in the context of like, you know, historical evolution, but particularly in the context of the epidermis, there is definitely keratin-specific domains that we think are the ones that are interacting with the granules.
Thank you. And the really last one, the student just followed up with the question about pH. Have you tried other external stimuli, assembly, disassembly besides pH?
Oh, that's a cool question. So that's a great question because I focus on pH, but because a lot of the biology that we've done really has to do with pH, we think it's an essential. It was actually unknown. We kind of uncovered that new pH transition, pH shift was unknown. So we kind of uncovered that. It's a pretty cool observation that was inspired by our work with phase separation and membraneless organelles. But you can imagine the skin is beautiful because the skin is kind of the exciting example where, although we typically think of cells as being homeothermic, maybe that's not so true for the epidermis, right? The epidermis is the interface with the environment. These organelles may be experiencing a bunch of different interesting environments. And so we think a lot about temperature. And we know temperature is a potent modulator of these phase separation dynamics. We've done a lot of experiments in the past and even in the context of keratoconine granules. And although we don't really know how temperature particularly contributes to the biology, I can tell you that the viscosity is shifted dramatically by temperature. And so we've done experiments where we subject these granules in the cell to different temperatures, and we see the shifting of the viscosity. So of course you can get, basically if you cool down, these are polymers that respond permanently to low temperatures. If you go from 32 Celsius to 15 Celsius, that permanently gels, kind of takes the granules to a gel-like state. And so we're very intrigued to explore how this shifting of the material properties in a pH-dependent, in a temperature-dependent manner might be contributing to skin biology, because in fact, people with skin barrier disorders suffer a ton when they go to cold environments. And so there's a lot of interesting implications with other stimuli beyond pH, and we're definitely interested in exploring those. And the skin is a beautiful system to do that.
Perfect. Thank you very much, Dr. Quiroz. Thank you for your talk. It was very, very, very interesting. So we're going to move on to